Hi everyone, welcome to the British Journal of Pharmacology article interview series. I am Shivani Sachdev, an editorial intern at the British Journal of Pharmacology and a postdoctoral fellow at NIH. So today we have David Gloriam with us. Can you tell us a little bit about your career path?
I got my master in pharmaceutical sciences and my PhD from Uppsala University. After that, I spent a fantastic year at the EMBL European Bioinformatics Institute outside of Cambridge, another year in industry with GlaxoSmithKline, where I picked up computational drug design. And from 2008, I've been working at the University of Copenhagen, where I have my own research group.
Yeah, awesome. So British Journal of Pharmacology with the Nomenclature Committee of the International Union of Pharmacology has recently published guidelines for design and reporting biased ligands experiment. Tell us more about how you constructed these guidelines. And since you are the senior author of this paper, I think our audience would love to hear directly from you.
Absolutely. Yes. So I think many people in the field have known for quite some time now there was a big need for community guidelines to, for bias signaling, for many reasons, because data has been a little bit unclear. And I think there's been some misunderstandings, both in how to be clear on what it is, but also how to do the experiments. So I remember speaking to pharmacologists at conferences. One of them were Arthur Christopoulos, who said some years back they had the idea to make a conference, a meeting where they would discuss this, but then it didn't materialize. What we did early on was trying to engage people. During COVID times, it couldn't be a physical meeting, but we reached out to invite some of the key pharmacologists to contribute to these guidelines. We had a big online meeting with many people initially, setting the outline and the direction. And after that, we had numerous one-to-one or just a few persons in the meetings. And I've been fortunate enough to lead this together with two other persons, one being Terry Kinaking, who is really a guru in bias signaling and expert in mathematical modeling, and the other one being Peter Kolb. Peter and myself, we are computational people, so why should we do guidelines? But we found out it was perfect because we wanted to make guidelines that everyone could understand, not just the expert pharmacologists. And also we were pretty good at structuring the different sections of the guidelines. We are data nerds, so we love to make a clear structure.
Yeah, that sounds like an interdisciplinary collaboration, and I like how Zoom has made our life easier, so I can see authors from everywhere coming together to construct these guidelines, and I think that's amazing. Bias signaling comprises very complex pharmacology, often making experiment designs and interpretation challenging. So how do you think we can avoid different type of bias, such as observational bias, which is the assay type specific, and then we have system bias, which is cell type specific, or it depends on the differential receptor expressed in the cells. So how do you think we can plan our experiments to avoid this type of bias?
Yeah, thank you for bringing that up. So our community guidelines have focused on ligand bias, which is, of course, the signaling bias that is due to the ligand only. And in trying to, measure that, you must isolate it from the other types of biases. And you mentioned observational bias, which is basically artifacts from incorrect experimental setups, and system bias, which is all other molecules in your system, be it the cells, the receptors, the concentrations of signaling proteins in the cell. So, the guidelines, they offer a number of experimental recommendations that you can do to isolate them in this way. Of course, ligand bias, it compares the signaling of two different pathways. So one way, of course, to reduce the observational bias is to make sure that you have as similar systems as possible. so that you use a similar assay, the same cell line, the same reference ligand, that you keep as much as possible the same, and so that what's left and what you're really comparing is the ligand.
Mm-hmm. Yeah. Yeah, I think I found that Appendix A, which is like experiment recommendation checklist, has
Yes.
like everything. And if you do not have time to read the entire paper, just go through the appendix section, and that would give you so much information.
I agree. Maybe we should have put that first in the paper, but these appendices,
Right.
it's really good. So just post them on the wall in your offices of your PhD students. So,
yes,
because they
definitely.
give a very nice quick overview of the experimental guidelines and the reporting guidelines.
Yeah, I agree. Yeah. So that takes me to my next question, which is bias cannot be reported without the use of reference ligand. What are some of the factors that should be taken into consideration when choosing a reference ligand for bias quantification?
Yes, so I would say this is maybe the most important consideration, and it's also one of the things that maybe people haven't thought enough about, and this is what leads to some of the confusion. So we set out in the guidelines to define 3 types of bias, which is defined by 3 types of reference ligands.
Right.
So I think the first one is what we call pathway bias, which means the isolated or at least strongly preferential signaling of your tested ligand in one out of 2 pathways, such that you can get a functionally selective response. And for that, you need to use a reference ligand that is balanced in these 2 pathways. So it has about a near equal signaling. And the guidelines describe how you can define the balanced reference ligand, for example, by using a so-called bias plot. The second type of bias is the physiology bias. It uses as reference an endogenous ligand. And for that reason, the meaning of physiology bias is that your signaling differs from the physiological signaling. Yeah. But bear in mind, the endogenous ligand may not be balanced. It may be pathway biased in itself, and therefore a ligand that is found to be physiology biased may actually be balanced across 2 compared pathways. So very important distinction. And finally, the third type of bias is what we call benchmark bias. It doesn't convey pathway bias or physiology bias. It merely compares a difference relative to the reference, which is often interesting drug or a highly used tool compound. So what you're really looking for is how your tested ligand differs from this drug or from the tool compound.
Yeah, and I also read in the paper that maybe sometimes we don't have reference ligand for the bias quantification. And in that case, there's a term called pathway preference. And you could calculate it using log Emax over EC50. And you could have like rank order of the ligands without defining a fixed reference point. So in the paper,
That's absolutely correct. If you don't have a reference ligand, it's no longer bias what you're doing. But what you can do is you can compare one ligand without the reference across pathways, and this is what we call pathway preference. It can be useful to select which ligands you should later test in a bias experiment. It can also be used to select a so-called pathway balanced reference ligand. So you identify the reference ligand you can use to test other ligands and their bias.
Right. In the paper, you also discussed about how 2 pathways measured at different depth can be problematic. For example, arrestin is almost nearly always measured as recruitment or translocation to the membrane, while G proteins are measured or often measured at the downstream level, like cyclic AMP production for GS proteins. So how do you think we can minimize SA amplification effects that can confound efforts to identify truly biased ligands?
Yeah, absolutely. So yes, as you said yourself, you should try to measure the signaling at the same depth. So for example, recent so-called breadth assays, many of them measure the recruitment of G proteins or arrestin to the receptor. Related assays sometimes measure the dissociation of the G protein. These assays are close to the receptor, they have little amplification, and therefore they may be more consistent in your comparisons. But unavoidably, sometimes you can't measure anything else than a downstream signal. And sometimes the downstream signal is maybe closer to the physiological effect that you want to measure. Again, just try to maybe go as far downstream in both pathways, and also to make sure that you use a reference ligand that has a near equal signaling if you're looking in pathway bias.
Okay, yeah, no, that is a really good advice. So what is the best way to quantify ligand bias for the tricky ligands? For example, ligands that are dead in one pathway, but they may activate the other pathway with a very high efficacy.
Yeah, that's right. So there are several types of tricky ligands. Some are the ones that don't have a full efficacy, meaning a full activation in one of the pathways. So they're partial agonists. So if the reference ligand, if there doesn't exist a reference ligand for bias that is both pathway balanced and has full efficacy, you can actually use two reference ligands in the same experiment. So you use one reference ligand for the bias, but you also use another reference ligand for the efficacy. And then you compare both the reference ligand for bias as well as the tested ligands against the same reference ligand for efficacy. So that's one way to deal with that. Another type of tricky ligands would be ligands that stimulate, agonize one pathway, but they are neutral antagonists in the other pathway, giving no signaling. So in this case, you cannot calculate the quantitative bias, but you can calculate, for example, rank orders. There's also what you say inverse modality, where you maybe inhibit one pathway and stimulate another. And this is what we refer to as modality bias in the guidelines. So you can get a full differentiation of the effect, but you do it via the modality of the ligand.
Wow. Yeah. And I think now that leads me to the last question of the interview. And as we all know, there's some controversy regarding the importance of bias and its druggability. What are your opinions on this?
Yes, that's right. So, for example, people have been looking at Trevena's compound, eliciting, that was approved by the FDA in 2020. For those who don't know it, it targets the mu-opioid receptor, and the bias rationale tried to avoid the lethal breathing suppression through bias signaling. It was later found that this hypothesis didn't hold up. I mean, the drug was approved, luckily, but the bias rationale didn't hold up. I've been to recent conferences where this rationale has been updated. So, for example, I've listened to Brian Kobilka present that what he thinks needs to be done to avoid the breathing suppression is to get bias, but not between G proteins and arrestin, but within the same G protein family, specifically the GIO family, where you would get a ligand activated some of the subtypes of G proteins, but not the others. So I think that is very exciting. Something else that is very exciting is that last month, there were 2 papers published where the group of Brian Choquette has done virtual screens to identify new ligands with bias, both targeting the mu-opioid receptor for analgesia, as well as another receptor, and in both cases achieved biased drug candidates from virtual screening. So this is a first proof of concept that you can use computational approaches to quickly identify ligands that have close to drug properties.
Yeah, I agree with you. I think there are a lot of papers coming from their lab about the bias signaling profile for the opioid receptor as well as for the serotonin receptor, I think. Yeah, and I completely agree with you that but that it's a long way from bench to the bedside because there's so much we don't know about the physiological consequence of the pathways. And I feel like disease is not just one pathway. It's like combination of different pathway that leads to some disease. And I guess part of the problem is also reproducibility and inconsistency in cross-lab characterization of this ligand. I think which is where this paper comes in, and it provides you do's and don'ts of bias ligand quantification. And I'm sure that readers and those who work in the field will feel the value and benefit of this article. And David, is anything else you want to add?
Yeah, now that you mention it, how different labs come to different conclusions, I think maybe different labs can also help each other out. So one example is where one group may publish initial in vitro assessment of the bias of some ligands. Other groups could follow up on that and use more advanced assays and maybe in vivo systems. And towards that, my group has recently submitted a paper describing a biased signaling atlas. You don't need to wait for the paper because you can find the atlas online. You can go to the page of GPCR DB database. You'll also find the biased signaling atlas next to it. And in there, you will find a very comprehensive annotation of biased ligands from literature. So in this way, we'll be able to go from one group, maybe to another group, following up on those ligands and validating them in another system, helping reproducibility.
Yeah, definitely. And thanks for your contribution to the field. I think I love GPCR DP, and I feel like it's great that you have everything in one place. If I am working on a new GPCR or something, I'll just go in there, look for the ligands, receptor, and the downstream effector, and it's just all in one place. Like, it's so impactful and productive for everyone in GPCR field. So yeah, with that, thank you so much, David, for taking the time to talk to us. I really enjoyed our conversation.
Thank you.
Please click on the link in this video to read the full paper. Thank you.